So we left off last time looking at somatic cells and this idea that you get two copies of every single chromosome. So you got one from mom inside the egg cell and one from dad inside the sperm. Now we actually have a term for this, and this is kind of part of the vocab introduction that you'll see a lot of. Somatic cells are diploid. Remember, di as a prefix means two, and ploidy refers to the number of copies of a chromosome. So diploid literally means two copies. So you have one copy from mom, one copy from dad, gives you a total of two. We can abbreviate this, and it often is abbreviated as 2n, where n is a variable referring to the number of copies of a chromosome. So the number of copies of a piece of genetic information. Now, an interesting kind of term that we want to add into this is this idea of homologous pairs. So homo as a prefix means same, and logos or loci means location. So a homologous pair means same location. Now, what this essentially means and where we get that term from is the idea that we are going to have two copies of a chromosome that has the same exact genetic information in it in the same place, but they are not genetically identical. So this is your mom version and your dad version of the same chromosome. So if we go back to one slide really quickly, I'm going to pick on chromosome number 1 right here. So here's my mom copy of my chromosome. Here's my dad copy of the chromosome. Now what we're going to find is that they'll have the same genes, the same information in the same place. We'll go to our next diagram to look at that one. So let's say on my mom copy and my dad copy at the top up here, I have a section of A's, T's, G's, and C's that codes for eye color. Now it's the same location for this eye color gene, but my mom might have given me eye color information for making blue eyes, and my dad could have given me information for making brown eyes. So see, they're not genetically identical, but it's the same gene in the same place. And those are called homologous pairs. So your mom and dad copy of the same chromosome. Now, mitosis for us is going to be the process of actually looking at how eukaryotic cells take these diploid somatic cells. So these cells in our body that have two copies, a mom and a dad copy of every chromosome, and make an exact copy of them. So this was where I started with this idea of you have a liver cell doing its job, but I want to make two new liver cells to replace that one that's going to die. So I need to make exact copies. And that's what mitosis will show us how that happens inside of a living thing. The next type of cell that we're going to tackle are your sex cells or your gametes. So gametes, your sperm and egg, they are haploid, meaning they only have one copy of each chromosome. Now we can quickly realize why this is a very good thing. So in order for humans to maintain the number of chromosomes, so humans have 46 chromosomes. When we reproduce, if we did not cut our number of chromosomes in half, every single generation would double the number of chromosomes. And this would lead to basically reproduction ending very, very quickly because you'd have a tremendous amount of genetic information and tremendous confusion within the cell as far as what to make and what not to make. So what we have is the system by which our cells are going to create these special cells in their body. So gametes, either sperm if you're a male, egg if you're a female, and these are going to have just one copy of every chromosome in it. So what can happen during fertilization, where the sperm and the egg combine to make that first cell, that zygote, is that zygote will have exactly the same number of chromosomes that mom and dad did. So we can maintain the number of chromosomes between every generation by cutting them in half in our sex cells. And the abbreviation for this, because it's just one copy, is just N. Now, what we're going to study in this unit is we're going to look at meiosis. So meiosis is the process by which eukaryotic sexually reproducing organisms are going to create these haploid gametes. So we're going to look at what type of division leads to sperm cells, what type of division leads to egg cells, and we'll examine that process in depth. So I've been showing you guys this picture a lot. This is the karyotype, and I've been saying this is a mom chromosome, this is a dad chromosome. And some of you might have just looked at it, taken it for granted, but you might have had the idea across your head, no, Miss Curley, chromosomes look like X's. And the interesting thing about this is that you are not wrong. There are actually two forms of a chromosome. So chromosomes that you are used to thinking of. So when you picture in your head a chromosome and it looks like an X shape, what you are actually picturing is something called a duplicated chromosome. So a duplicated chromosome is going to be found in a cell that's preparing to divide. So it doesn't matter whether it's going to do mitosis or meiosis. If it's getting ready to split itself at all, the first step of that is always copying its chromosomes. So a chromosome that is not getting ready to divide or has just finished dividing is going to have chromosomes that are linear, so just straight lines, like the ones we've been seeing in our karyotypes right here. So this is a cell that has just finished dividing. It's not getting ready to. And it's got one copy of every single gene on it. So you notice you've got a dominant A, a recessive B, and a dominant C. What's going to happen is there's a special stage in a cell's life where it makes an exact copy of every chromosome. And those are going to attach to each other at this point called the centromere. So the centromere is that point of attachment between my two duplicated chromosomes. Now notice that my two halves of a duplicated chromosome are exactly genetically identical. So I was dominant A, recessive B, dominant C, dominant C. And that's what you should see if there's no mutations inside of a duplicated chromosome. One more layer onto this. So we actually have a term for the two halves of a of a duplicated chromosome. We call each half of a duplicated chromosome a sister chromatid. And again, sister chromatids are genetically identical to each other. And what we're actually going to do in all types of cell division is we're going to split sister chromatids. So a cell should never end division with still having X-shaped chromosomes. They always split. So to kind of go right to the heart of the matter, this is something that tends to confuse a lot of students. What's the difference between a homologous chromosome and a sister chromatid? So remember, going back to the karyotype, we said that in diploid cells, di meaning two, you got a mom copy and a dad copy of a chromosome. So the mom copy and the dad copy are your homologous chromosomes. They have the same genes on them in the same place, but again, mom and dad aren't genetically identical twins, meaning that they're going to have different information in that space. When your cell copies itself, my mom copy makes an exact copy of itself and attaches at the centromere, and here's my duplicated chromosome made up of two sister chromatids. My dad copy of the chromosome, it makes an exact copy of itself, attaches at the centromere, and here's my duplicated dad chromosome made up of two identical sister chromatids. Sister chromatids, genetically identical. Homologous chromosomes, same genes, but not genetically identical. We'll talk about these quite a bit more.